Greetings to you all. This is teacher Dr. Mgomba Ronald. Today we are going to go ahead and talk about cell structure and function. That is what we are going to basically be looking at. In this chapter, of course, we are going to be looking at the different structures that are found within the cell and their different functions. This specific chapter is going to be in two videos. Part 1 will first go ahead and talk about the very first part of the cell and the different structures of the cell, and then part 2 will talk about the other structures of the cell that we will have left out and the movement in and out of the cell or cell physiology. So to begin with, when you look at the different types of cells as observed here, you can see we have a number of cells as viewed under the light microscope, like the onion cell, the onion epidermal cell. You can clearly see that. Then you can go ahead and see, like this is onion epidermal cell. You can clearly see it. Nasal sinusal cells, you can clearly see them. You can see even the cell of vibrio, which is clearly seen here. So when we are studying the cell, most of the times, because the cell is microscopic, we cannot see them with our naked eyes. We usually use a microscope when we are studying the cell. Studying cells, usually we use microscope. And the size upon which they do magnify, the electron microscope magnifies images to as a maximum of 10 million. The light microscope, which is the commonest that we use, to a maximum of 1000 magnification. The dissecting microscope to a maximum of 100 magnification. So you can clearly see that most microscopes that are used in our labs are usually cells up to approximately 400 times. So you can clearly see now this one here, this one here is the common light microscope, and then this one here is known as the dissecting microscope. The dissecting microscope usually has a very lower magnifying power, and are usually used to observe bigger objects like an insect. You can easily see it there, bigger objects. Now, when you look at for instance, when you are to look at the images that are produced by the electron microscope and the light microscope, you can clearly see the difference. That the electron microscope, the microscope that usually relies on a beam of electrons which are usually projected through the specimen to be studied, and then an image is formed on the film. This light microscope, it is different. For it relies on light. So you can see most of the times we use the microscope for checking out different organisms that might be found in our blood. For instance, when you are looking out for Plasmodium parasites, for those of you who have gone to hospitals, you must have noticed the fact that they take your blood and they make a blood smear, and then they go ahead and check whether you have the Plasmodium parasite. So like here we have the cells of the cervix. That's how they look. So when you look at the cell, we are going to start by looking at what they call the cell theory. The cell theory, as proposed by Robert Hooke in 1665, he's the one who initially discovered the cells when he sliced pieces of cork. And then a man known as Matthias Schkelden in 1838 is the one who went ahead and, along with Theodor Swan in 1839, they proposed the cell theory, and it was modified by a man known as Rudolf Virchow in 1858. So the modern ideas of the cell theory. So cell theory, basically the cells are the basic unit of life, and all the different chemical processes that take place within all living things start from the cell level. So that is basically the cell theory. For it talks about the cell being the basic functional unit of life, and all the different life processes begin at the cell level. And it proposes as well that all cells arise from pre-existing cells. So cells divide by a process known as cell division, and they form new cells from the pre-existing cells. And also, cells are a building block for all living organisms, whether it is single-celled like these ones which are single-celled, that is unicellular. Those organisms which are single-celled like some algae, that is single-celled, it is the whole organism is made up of only one cell. Then we have those ones which are known as multicellular organisms, whose body is made up of very many cells, like most of the plants and animals. Each type of cells with a specific function. We shall look at that in our next, we shall look at it in future. When you look at each of those cells, they are specialized to perform specific jobs. Different cells within the body of a plant, within the body of an animal are specialized to perform very many different functions. Like the immune cells, like an example that we can give is the lymphocytes, the T lymphocytes, the macrophages, the B lymphocytes. Those are all immune cells which are very important when it comes to talking about their function. They are very important when it comes to defending our bodies against diseases. When you look at the different, before we can go ahead and look at the cell theory, we should, after we have looked at the different aspects of the cell theory, allow me to take you through some of the exceptions. I've not put them on the slideshow, but I believe if I talk about them, you are most likely to reflect about them. There are exceptions to the cell theory, and there are some living organisms that go against the cell theory. An example are the viruses. When you look at the viruses, for them, viruses are oblique intracellular parasites. That means that obligate intracellular parasites. This means that they cannot survive when they are outside the cell of an organism. They reproduce within the cell of an organism. For them, so what should we say? Are they biotic or abiotic? Because they are not cellular, they are not made up of cells. Then we also have another exception that is observed in the cynocytic algae, like Ventureria and very many different algae, which have their bodies that are usually contain a continuous mass of protoplasm. And you find that within the protoplasm, there are very many nuclei found within the same protoplasm, without any cell wall septa or receptor that is separating the different protoplasms into independent cells. Look at another exception comes from the skeletal muscles. When you look at the skeletal muscles, they have very long cells with very many nucleus. When you look at the giant algae, it's an exception. It's one cell, but it is very long, which can even go up to 100 millimeters in length, but only has one nucleus. Look at the unicellular organisms that can be considered as cellular because they are larger than a typical cell. There are some organisms which are unicellular and they are far much larger than a typical cell, and so on and so forth. So those are some of the exceptions of the cell theory. Talking about viruses, cynocytic algae, skeletal muscles, giant algae, and unicellular organisms. Now, when you go ahead and look at the different cell types, we have a number of different cell types. We have very many different, we have basically, the cells are divided into basically two types of cells. However, we need to first go ahead and talk about the cell components. When you look at the cell components, we have the cell membrane, which forms a boundary. It's like the fence or the wall fence that is surrounding the cells. The way we have wall fences around our homes. The cytoplasm is the one that is liquid, and it is the one that contains the cell organelles and the different inclusions, as we shall see. The nucleus is the one that contains the DNA. Now, when you look at the different cell types, we have a number of different cell types. We have basically two of them. And to mention but a few, we have the prokaryotic cells, that is to say the bacteria. The word prokaryotic comes from two words, pro and karyon. Pro meaning before, and karyon means true nucleus. So organisms that evolved before the evolution of the nucleus are known as the prokaryotes. And then the eukaryotes are those organisms which have a true nucleus. Like the plant cells, animal cells, their nucleus is, they have their DNA enclosed within the nuclear envelope. Unlike the prokaryotes like the cyanobacteria and the bacteria, for them they are even very much smaller. in size, far much smaller in size, like 10 times smaller than the eukaryotes. When you look at them, they do not, their DNA is naked. The prokaryotes, their DNA is naked, unlike for the eukaryotes, whose DNA is enclosed within the nucleus. When even you look at other structures that are found within the prokaryotes and eukaryotes, they're different. When you look at, let's say, the cell wall that is found within the eukaryotes, especially in the plants, they contain cellulose. And when it comes to the fungi, they contain chitin, and then in animals like insects, they contain chitin as well. Now, when it comes to the bacteria, for them, their cellulose are usually made up of a complex molecule known as peptidoglycan. Peptidoglycan is the molecule that makes them up. When you look at them specifically, when you look at even the type of ribosomes, the eukaryotes usually have the 70S ribosomes and the 80S ribosomes. But when you look at the prokaryotes, they only have one type, that is the 70S ribosomes. Now, the prokaryotes they lack a nucleolus, but the eukaryotes have a nucleolus. Putting that aside, so you can look at the structure of a bacterium. You can clearly see most of the eukaryotic characteristics being showed here. You can clearly see that the DNA is just spread into the cytoplasm. It is naked, it is not enclosed, and their DNA is known as the circular DNA. And in comparison to the eukaryotic DNA, which is linear DNA, for them they have a circular DNA. Their cell walls are made up of a complex molecule known as peptidoglycan. Their cell membrane is usually having no carbohydrates, and even it lacks the sterols. It doesn't have carbohydrates, and it does not have the sterols also. Unlike when it comes to the cell membrane of the eukaryotic cells, which also have carbohydrates and sterols within them, the sterols. When you look at the ribosomes, I've talked about them. The prokaryotes have 70S ribosomes, whereas the eukaryotes have both 70S and 80S ribosomes. You can see a flagellum is not for all of them. Some of them have it, others don't have it. They have a capsule. Then we have the pili. You can clearly see that. Now, you can see the different cell sizes. When you look at this carefully, this figure is showing the relative sizes of different kinds of cells in unicellular and cellular components. An adult human being is shown in comparison. So you can see the different cell sizes, and you can clearly notice that the bacterium is among the very small ones here. When you look at it carefully, you realize that the bacteria is among the small ones, very small. It is almost a similar size as the mitochondria. And this explains how the mitochondria, by the way, as we shall see later, how the mitochondria came into existence from, they say, it was a pre-living prokaryote and which went under evolution and then became engulfed by the big bacteria, and it gave rise to the mitochondria within the eukaryotic cells. So you can clearly see the comparison between the cells. That process, that theory that tries to explain the rise of the eukaryotes from the prokaryotes was proposed by a person known as Lynn Maglias in 1967. She proposed the theory known as the endosymbiotic theory, and I would urge all of us to go ahead and read about that theory of the endosymbiotic theory. Endosymbiotic theory. So, now when you look at the endosymbiotic theory as proposed by this lady in 1967, it suggests that mitochondria and the chloroplasts were once separately existing small microorganisms, the aerobic bacteria and the photosynthetic bacteria. And it is said that there were two types of bacteria, the big bacteria and the small bacteria, which the big bacteria was moving around, and it came across this aerobic bacteria that could easily carry out its own metabolism and produce energy. It engulfed it. When it engulfed it, it swallowed it but did not digest it. When it remained within, that is why they call it endosymbiotic. It means you take it in, but you do not digest it. You get an association. You get into an association with the two. The two get into an association. They live together harmoniously. So the aerobic bacteria became the present-day mitochondria, and it started carrying out metabolism for this big bacteria, producing energy. So it was non-digested. Now, when it came to the big bacteria was still moving around, and it came across the photosynthetic small bacteria, which would carry out normal photosynthesis, breaking down using carbon dioxide and water to form carbohydrates for it to survive. So these bacteria went ahead and also engulfed it. As you can see it here, it engulfed it. So when it engulfed it here in this illustration, you can see it here. When it engulfed it, so you can see it also engulfed it. When it engulfed it, it was taken into the cell. When it entered within the cell, it did not digest it, but instead it retained it, and it remained within the body of that big bacteria, and it became the chloroplast. And then when you look at, we have a number of reasons as to why. When you look at there are similarities. When you look at these people who proposed the endosymbiotic theory to try to explain how the rise of the present mitochondria and the chloroplast, they defended their argument by looking at the different features that are common in the prokaryotes and are also common to these cell organelles. One of them is when you look at the DNA. When you look at the prokaryotic DNA, it is a single circular chromosome. Yet the eukaryotic DNA is linear and chromosomal. But when you look at the mitochondrial DNA and the chloroplast DNA, it's also circular, and so that is enough to explain that. To show that the mitochondria and the chloroplast came from the free-living prokaryotes. When you look at duplication, you realize that the prokaryotes duplicate by binary fusion. Eukaryotes duplicate by mitosis. But when you look at the mitochondria and the chloroplast, it's by binary fusion. When you look at the types of ribosomes they have, you will all appreciate with me the fact that the mitochondria has a 70S ribosome along with the chloroplast, similar to most of the prokaryotes. Now, when you look at the eukaryotes, they have 80S, and so on and so forth. So that is basically the endosymbiotic theory. It was proposed in 1967. So we are going to go ahead and also look at the different components of the cell. And we are going to go ahead and look at the eukaryotic cell and the different components. So this is an overview. When you look at the different components of an eukaryotic cell is made up of the cell membrane. The eukaryotic cell is also made up of cell wall for plants and yeasts and also fungi. When you look at it has a nucleus and the cytoplasm. And within the nucleus, within the cytoplasm, you will find membrane-bound organelles known as the mitochondria, the ribosomes, endoplasmic reticulum, Golgi apparatus, lysosomes, peroxisomes, cytoskeletons, vacuoles in plants, and chloroplasts also in plants. So we are going to look at one by one, and we see each of them. So when you look at a typical plant cell and a typical animal cell, in this slide, you will realize that there are a number of features that we shall talk about. So we are going to go ahead and look at one by one. So this figure here is showing a typical plant cell, a typical animal cell, and a typical plant cell. So when you go ahead and look at the cell membrane, we are going to go ahead and look at the cell membrane. The components of the cell membrane, when you look at the cell membrane, it is predominantly lipid. It is made up of predominantly lipids and proteins. Though it also has, it is made up of predominantly lipids and proteins, though it also has carbohydrate. So when you look at the lipids have been organized in what we call a phospholipid bilayer. The word phospholipid means the molecule that is here is a phospholipid. It has a phosphate head and a lipid tail. Now, and we have also cholesterol that is interspersed between them. We are going to see it. The proteins are arranged in a mosaic pattern. The proteins, they are arranged in the membrane in a mosaic pattern, interspersed all through the cell membrane in an irregular pattern. The carbohydrates, we shall see them, they are usually attached to the proteins or the lipids. Then we are going to go ahead and look at the different functions of the cell membrane. When you look at the cell membrane performs very many different functions, as we are going to go ahead and look at. And we are going to look at the different functions of the cell membrane. We are going to go ahead and look at the different functions of the cell membrane, but one of them is the fact that it allows selective passage of materials in and out of the. So you can clearly see the different parts that make up the cell membrane. We can go ahead and look at the description. I've told you, when you look at the cell membrane, it is a plasma membrane. Sometimes it's called a plasma membrane. You can see clearly the phospholipids. They have a phosphate head projecting outwards to the aqueous cell surrounding and inwards to the aqueous. So when you look at the different parts components of the cell membrane, you can see that the heads are projecting outwards in the bilayer because they are water-loving. They are known as hydrophilic, and they project outwards into the aqueous surrounding of the cell and the aqueous cytoplasm of the cell. And then when you look at the tails for them, the tails basically project inwards towards each other, and they form what we call the hydrophobic region within the cell within the bilayer. That liquid area here is called the hydrophobic region in between these two, because it is made up of fatty acid tails which are water hating, so they project inwards and they form a hydrophobic region. So you can see that some proteins form what we call the channel proteins. You can see it here. This is a channel protein. Other proteins remain attached to the inside, that is the peripheral proteins. Others on the surface of the membrane, peripheral proteins. Now, some of the proteins get attached to small fragments of oligosaccharides. They form glycoproteins. Others, the lipids, sometimes they also get attached to parts of oligosaccharides and they form glycolipids. You can clearly see that. You can see the filaments of the cytoskeleton. So the cytoskeleton of the cell is usually composed. So before we can go ahead and look at the cytoskeleton, allow me to first go ahead and take you through the different functions of the cell membrane, depending on the different functions, the different components of the cell membrane and their functions. So when you look at the cell membrane and their when you look at the components of the cell membrane, you realize that we have very many of them. Like the proteins are very important, acting as antigens. When you look at the proteins, can act as channel proteins to allow diffusion of polar ions to go through. The proteins can sometimes act as transport mechanisms that will allow materials to cross the cell membrane. The proteins can also act as cytoskeleton to provide shape to the cell. The membrane proteins can also form junctions between the cells, as we are going to see them. When you look at some cell membrane proteins become enzymes which are involved in different reactions in the cell, like the process of the electron transport system. The glycoproteins are very important in cell-to-cell recognition, how cells recognize each other during the process of development. The cholesterol is very important in stabilizing the cell, and the lipid bilayer is also very, very, very important because it is semi-permeable to the different molecules. So today we have had an introduction. We have had an introduction to the cell. Now, in the next video, we are going to go ahead and look at different cell organelles that are found within the cell, one by one, structure and function. I remain teacher Dr. Mugomba Ronald. Consider to subscribe, like, share, and comment about the different things that you have come across in the video. I remain teacher Dr. Mugomba Ronald, MBCHB.